There are estimated to be 5,000 to 6,000 viruses ready to jump from animals to humans, and an additional 1.5 million unknown viruses out there. The risk of infectious disease is growing as humans increasingly encroach upon the natural environment. These viruses threaten us with the potential of future pandemics, many of which may be more lethal than any we've seen before. We can prevent this.
200 years ago, humans invented vaccines to combat viruses by killing them or preventing their entry into cells. As we speak, there is a new cutting-edge technology being developed, the mRNA vaccine. mRNA is a copy of DNA that takes genetic information to be made into proteins. When mRNA is injected as a vaccine, our cells use it to make the protein. Our immune systems learn to recognize the protein as an enemy part, and when the real virus enters our bodies, our defenses will be primed and ready. Moderna, among other biotech companies, is developing the first mRNA vaccine with mRNA that codes for the SARS-CoV-2 viral spike protein, and it has passed initial clinical trials with promising results. Conventional vaccines usually take 10 to 15 years to produce, but mRNA vaccines could be ready in a fraction of the time.
Science, engineering, and design have always drawn inspiration from biology and the natural world. Every organism on Earth is constantly evolving as they compete for resources in an evolutionary arms race. But what if we were able to look into the future and predict these evolutionary changes? Imagine the lives we could have saved if we had predicted what was coming. If we'd had policies already in place, vaccines ready to be distributed, and no need for a global lockdown at all. Just as we built optics inspired by butterfly wings and neural networks inspired by human brains, we can create software inspired by evolution. This is Viper, a biology-informed software tool designed to simulate the evolution of viruses.
How is predicting the future possible with Viper? There has been a lot of effort trying to model SARS-CoV-2 evolution using traditional bioinformatic techniques. We would like to challenge ourselves and take on a novel path of using deep learning combined with time series regression to model and predict how spike proteins will look like in the coming months. To start, we analyze spike protein diversity and phylogenetic relationships for gaining a better understanding on protein evolution. From GenBank, we download more than 9,000 sequences and their associated metadata. To review protein diversity, we studied mutations and their frequencies, identifying mutations impacting protein function, as well as revealing mutation hotspots and deserts. From there, we wanted to further our knowledge on amino acid substitutions happening on the spike protein, as they are the most frequent variant type given the short time frame. So we constructed a sequence profile where we determined the probability of each position along the spike protein being substituted by a specific amino acid. With this profile, we were able to generate a large number of mutants that are still representative of our dataset, which can then serve as a null model. Finally, we constructed a phylogenetic tree and grouped the sequences into evolutionary clades for later use. Next, we wanted to represent viral sequences in meaningful numerical formats, such that they can be interpreted by computers. Variational autoencoders, or VAEs, are deep learning models capable of compressing text data or images into meaningful lower dimension representations. In other words, we use VAEs to represent a spike protein sequence with numbers, and sequences that are similar to each other should be mapped to similar numbers. A VAE is made up of an encoder and a decoder. The encoder compresses our sequence data to its latent embedding. In our project, the number of latent dimensions was set to 3 to allow for easier visualization, and thus each sequence was compressed to 3 numbers. The decoder is then able to reconstruct sequences from the latent embedding coordinates. Since our current dataset doesn't have enough unique sequences to train VAE models properly, we simulated 20,000 mutants based on the sequence profile we analyzed in the previous step to build a training dataset for VAE. Training in VAE involves a supervised process where each spike sequence is fed into an encoder, which compresses it to 3 numbers. And the 3 numbers is then inputted to a decoder, which reconstructs a sequence. Then we compare the reconstructed one with the original one and generate a loss score. During training, the loss score should be decreasing. We used an early stopping function to stop training once the loss score has stopped improving, so that we can avoid overfeeding our model. Afterwards, we input our actual dataset to the encoder and get the numerical representation of each spike protein. Here we can visualize where the sequences are located in the 3-dimensional space. Each dot is a spike protein. Each axis represents the coordinate translated by an encoder, and the colors represent the relative time that the spike protein is detected. For later time series regression, we split our sequences into the ones that occurred prior to May 30th as another training set, and a testing set containing the ones occurred after May 30th. With numerical representations of the proteins, we can now move forward to model time series changes. For better illustration, we plotted each coordinate against time. The horizontal axis is time, and the vertical axis is the coordinate value produced by VAE. And we have one plot for each coordinate. To make our models more specific to an evolutionary path, we leverage the results from our phylogenetic analyses. Essentially, for each time series model, we only used sequences appearing on the same phylogenetic lineage. Since our dataset only contains a fraction of all of the viruses out there, the next step is to find out the underlying function that best model our data. The method we use for that is called Gaussian processes, or GP, a Bayesian statistical model for fitting a function to an observed dataset and generating predictions for unseen time points, given what has been observed. It involves a supervised learning process where we gift the observed data with their associated time points so that the model can estimate the best fit functions. Here is a figure showing the estimated functions from Gaussian processes. The purple lines in the figure show the regression functions from our model, and the green points are the actual data points we observed. Generally, the regression lines fit tightly around our data, meaning the Gaussian process can model the trajectory well. From the regression functions, we then asked the model to produce predictions on new time points. As a proof of concept, we sampled 1,000 predictions at 30 days after the last training time point. These data were then passed into the decoder in our VAE to translate back to amino acid sequences. Since numerical data from Gaussian processes are continuous and protein sequences are discrete, multiple predicted values could be mapped back to the same sequence, and hence each predicted sequence has an associated frequency. We then choose the most frequent sequence for further evaluations. Our next challenge was to assess how well our model was predicting spike protein sequences that would be prevalent at future points in time. We tried to tackle this issue from both the sequence and the structural level. On the sequence aspect, We compared predicted sequences with our previous diversity analyses and see if the predicted mutations happened on variant hotspots or deserts, or if the mutation effects are known from previous research. Mutants with unknown effects are of more interest to be a new vaccine target. On the structural level, we would like to further characterize protein activities. Since spike proteins play a major role in viral entry to human cells by interacting with the ACE2 receptor, we thus were interested to see how well our predicted sequences can bind to ACE2. While going about trying to determine this binding affinity, we used many different computational tools as well as spoke with many different experts. We came up with a plan on how we could assess the binding affinity. First, we determined the three-dimensional structure of a mutant spike protein with homology modeling software such as Swiss model. Homology modeling is the process of constructing an atomic resolution, three-dimensional model of an amino acid sequence using an existing crystal structure of a similar protein. For us, we were able to use the crystal structure of the spike protein as a template and our model's output as the sequence. Next, protein docking will be performed using the three-dimensional constructed model of the mutant spike protein and the ACE2 receptor using docking software such as Rosetta. In the future, we plan to use a e-surface display platform and fluorescence-activated cell sorting to quantify the expression of our predicted sequences and their binding affinities relative to the wild-type SARS-CoV-2. This way, we could validate in silico fitness scores we obtained using the Viper software using in vitro experimental approach.
So, what can we do with this information? Recall that mRNA vaccines are based off of the viral spike protein sequence. If we can predict this sequence before the virus ever emerges, then we can begin vaccine development in advance. The wait time for a vaccine can be devastating, and this is what the world is seeing right now. And this could be even worse in a pandemic caused by a more lethal virus. Instead of waiting for a high-caliber, pandemic-level virus to strike, we can begin vaccine research way in advance. mRNA vaccine development is estimated to cost between $100 and $200 million, which is really a drop in the ocean compared to the estimated $4.2 trillion lost so far in the United States alone. This is a wake-up call. We need to begin investing more in preventative biomedical technology. Viper is our team's solution.
But could Viper's predictions lead to lost resources through development of unnecessary vaccines? Extensive consideration goes into developing each and every vaccine by virologists and policymakers alike to prevent the creation of vaccines that are not needed. And even if a predicted virus never arises, the cost of continued preemptive vaccine production would still be less than the cost of a single pandemic.
With the outreach component of human practices, with the outbreak of COVID-19 early this year, our team prepared some simple and informative infographics on the biology and basic ideas behind COVID-19, along with local resources for British Columbia residents. The Human Practices team has always worked to disseminate synthetic biology knowledge to various age groups, including a preschool audience. We're creating an illustration-based storybook, The Adventures of Kleb, that revolves around a day in the life of a bacteria named Kleb. The book aims to familiarize children with the idea of microorganisms, along with their potential benefits and harms, and the importance of hand washing to prevent infectious diseases. We also developed and hosted 3 bioinformatics workshops that introduced middle and high school students to the topics of genetics, bioinformatics applications and tools, and virology. The workshops also included hands-on coding activities like identifying an unknown DNA sequence using BLAST. We also had an activity that introduced the idea of recurrent neural networks and promoted understanding of the technology behind Viper in order to promote it as a learning tool.
The Human Practices team conducted interviews with first and second year university students to understand their level of knowledge about synthetic biology. We used this data to guide the development of a synthetic biology curriculum that aims to provide an overview of synthetic biology, its applications, as well as addressing misconceptions about this field of science in an easy-to-follow and interactive video format, which was most popular among the students we interviewed. To further our discussion about synthetic biology with the public, we're piloting a video series named Rewritten, in which team members introduce and discuss various synthetic biology concepts with people from various cultural and age groups, from elementary school children to expert researchers. Through this series, we aim to reframe the discussion around synthetic biology and demonstrate that scientific communication transcends the boundaries of age and culture. We believe it's important to educate the public about the science behind our cutting-edge technology so they can make informed choices about their health, such as vaccine implementation. Within the scope of integrated human practices, we believe another crucial aspect of every successful biomedical technology is continuous improvement. So our team is constantly striving to stay up to date in the field of synthetic biology and receive feedback from experts such as IT specialists, bioethicists, epidemiologists, financial strategists, and experts in machine learning. We also had a chance to connect with the research group led by Dr. Natalie Stranatka and Dr. Steve Slatter, who work on predicting the evolution of COVID-19 by using artificial intelligence, computer modeling, and structural biology. From them, we learned more about using machine learning and biological research and got more inspiration on pursuing computational technologies in our project. Also, they offered insights on how they assess sequence binding affinities in silico and in vitro, which guided our project development.
We sought expert advice from a bioethics professor who suggested that biased and insufficient data collection could lead to inaccurate predictions. These errors will be minimized as more viral samples are collected, leading to higher accuracy in our predictions. What about the potential unequal access to preemptive vaccines? Socioeconomic disparity is a global problem yet to be solved. An economics professor specializing in poverty analysis suggested working closely with local vaccination programs to ensure that the produced vaccines can be distributed to rural and vulnerable populations. Since the production of mRNA vaccines is 10 times cheaper than regular vaccines, this could be achieved under special health acts at a reduced cost or for free.
A vaccine to end this pandemic won't solve the root of the problem. The risk of infectious outbreak is increasing as we destabilize Earth's equilibrium. We intrude on ecosystems by cramming wildlife into unsanitary conditions, decimating habitats, and forcing wild species to come into closer contact with us. Additionally, climate change pushes disease carriers to expand their territory. All of these increase the risk of transmission between animals and humans. H1N1, Ebola, SARS-CoV-2, and 70% of all human pathogens originate from animals. Scientists expect new viruses to emerge and for old ones to return with a vengeance. Although Viper was trained on genetic sequences from SARS-CoV-2, its framework can be expanded to predict the evolution of other viruses as well.
Envision a world in which we diligently monitor virus populations in wildlife, livestock, and people. We could predict when, where, and which viruses will cause an outbreak, and have a vaccine ready before it happens. What if this world was within our reach? As we grow aware of the myriad of ways in which our actions impact everyone on Earth, it is time for humanity to unite with a collective goal, to use our innovation to influence the world positively with our actions. A new world is emerging. A world that prioritizes biology.